Welcome to the channel. This is the first biochemistry lecture and today's topic is gluconeogenesis. It's going to be the last lecture about glucose metabolism, then we are going to go through glycogen metabolism. In the previous lectures, we have studied the breakdown of glucose by glycolysis, link reaction, TCA cycle and etc., which all led to the production of ATP that will be used in energy requiring processes. Water and carbon dioxide were produced as waste products, and instead gluconeogenesis would lead to the formation of glucose. First of all, you need to know that the brain, RBCs, and exercising muscles require a continuous supply of glucose as metabolic fuel. Because glucose is the only carbohydrate that can be used by anaerobic glycolysis. During fasting, glycogen stores inside the liver supply their needs for less than 24 hours. After 24 hours, we call it prolonged fasting, when the cells must synthesize glucose from non-carbohydrate precursors. Gluconeogenesis is not the reversal of glycolysis because it enters special pathways. The equilibrium of glycolysis favors the pyruvate formation, and that's why gluconeogenesis needs to bypass the irreversible reactions of the glycolysis. Gluconeogenesis takes place inside the mitochondria and cytosol of the liver cells and kidney cells. Overnight fasting means sleeping, because during sleeping you do not eat, so you are fasting. Liver synthesizes 90% of the total glucose production, while the kidney will synthesize 10% of total glucose production. While during the prolonged fasting, kidney will synthesize 40% of glucose production. Remember, any deficiency in the enzymes of gluconeogenesis would cause hypoglycemia. Why? Because glucose is no longer supplied by diet. Glycogen stores inside the liver are depleted, so the only source for glucose is gluconeogenesis, and any deficiency in the enzymes involved in the gluconeogenesis would inhibit the process, and no glucose would be formed, decreasing the blood glucose level, which is known as hypoglycemia. From where could I get substrates for gluconeogenesis? Here are three sources: glycerol, lactate, and alpha keto acids. The first source for our substrates is glycerol, which is delivered by the blood to the liver cells after the hydrolysis of tags. Remember, tags are types of fats. Here, inside the liver cells, glycerol will be phosphorylated to glycerol 3-phosphate by glycerol kinase. Remember, 3 stands for the carbon atom to which the phosphate group is attached. Then, glycerol 3-phosphate will be converted into dihydroxyacetone phosphate by the help of the enzyme known as glycerol 3-phosphate dehydrogenase. Normally, during glycolysis, dihydroxyacetone phosphate would be isomerized to glyceraldehyde 3-phosphate. Instead, during gluconeogenesis, both of them are going to combine to give me fructose 1,6-bisphosphate that will be used by gluconeogenesis for the production of glucose. The second source is lactate, which is formed by anaerobic glycolysis that occurs inside the RBCs and exercising muscles. Then lactate is going to be transported by the blood to the liver, where it's reoxidized to pyruvate by Cori cycle. Again, pyruvate will be used by gluconeogenesis. Alpha keto acids During fasting, metabolism of all amino acids except lysine and leucine generate alpha keto acids such as pyruvate. Pyruvate will be converted to either glucose or alpha ketoglutarate, which enters the TCA cycle and forms oxaloacetate. Oxaloacetate is an important substrate for gluconeogenesis. Now let's go through the gluconeogenesis itself. It's made up of 11 reactions. 7 of them are the reversal of glycolysis, and the other 4 are alternative pathways. The first reaction, pyruvate is carboxylated by pyruvate carboxylase into oxaloacetate. Remember, pyruvate is made of 3 carbon atoms. Carboxylation means addition of 1 carbon atom. So this means oxaloacetate is made of 4 carbon atoms. This reaction is catalyzed by pyruvate carboxylase, which requires biotin as a coenzyme. ATP is hydrolyzed to produce energy required in the formation of oxaloacetate. The conversion of pyruvate to oxaloacetate occurs inside the kidney and liver cells, and it has 2 purposes. It gives me oxaloacetate, which is an intermediate for TCA cycle, and it would lead to the production of PEP that would be used for gluconeogenesis. Remember, TCA cycle and gluconeogenesis takes place inside the liver cells, because liver cells use PEP for gluconeogenesis and oxaloacetate for TCA cycle, while the muscle cells do not synthesize glucose. Instead, they use oxaloacetate for TCA cycle. This means gluconeogenesis does not occur in muscles. Pyruvate carboxylase is activated by the acetyl-CoA. Why? Because high concentrations of acetyl-CoA means increased synthesis of oxaloacetate is required. This is because acetyl-CoA and oxaloacetate are condensed together during TCA cycle. Then, oxaloacetate is reduced by the mitochondrial malate dehydrogenase into malate. What is the purpose of this step? We have to know that there are no oxaloacetate transporters inside the inner mitochondrial membrane, while there are transporters for malate. That's why we need to convert the oxaloacetate into malate, which can pass through the inner mitochondrial membrane to be transported into the cytosol. Then, inside the cytosol, malate would be reoxidized to oxaloacetate by the help of cytosolic malate dehydrogenase to give me oxaloacetate again. Then, oxaloacetate is decarboxylated and phosphorylated by an enzyme known as phosphenyl pyruvate carboxykinase. This reaction occurs inside the cytosol, and it is an energy requiring process. That's why GTP is hydrolyzed. Decarboxylation of oxaloacetate means oxaloacetate lost a carbon atom. Oxaloacetate was made of 4 carbon atoms, so PEP is made of 3. 4 minus 1 gives me 3, and it's phosphorylated. Now, after we got PEP, we would move into the opposite direction until getting fructose 1,6-bisphosphate. So we are going to go through in the opposite direction through all the reactions which was involved in the glycolysis. After reaching fructose 1,6-bisphosphate, we would face a problem. Which is the irreversible phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. Remember, inside the glycolysis, this irreversible reaction was phosphorylated or was catalyzed by phosphofructokinase 1. So we need to bypass this irreversible phosphorylation of fructose 6-phosphate to fructose 1,6-bisphosphate. It's irreversible, so we cannot go into the opposite direction. We bypass the irreversible reaction by the help of fructose 1,6-bisphosphatase inside the liver and kidney cells. Then, fructose 6-phosphate would be isomerized to glucose 6-phosphate. Remember, inside the glycolysis, isomerization of glucose 6-phosphate to fructose 6-phosphate was reversible. That's why we didn't face a problem. But after that, we would face a problem for conversion of glucose 6-phosphate to free glucose, because the phosphorylation of glucose to glucose 6-phosphate was irreversible. Again, here we need to bypass the irreversible reaction of hexokinase and glucokinase. Glucose 6-phosphatase enzyme would help us to bypass this reaction. And any deficiency in this enzyme would lead to hypoglycemia and glycogen storage diseases 1A and 1B. Remember, again, this reaction takes place inside the liver and not the muscles. Finally, after getting free glucose, this free glucose will be released into the blood to restore glucose level. Of course, as glycolysis and TCA cycles needed to be regulated, gluconeogenesis also requires to be regulated. First of all, glucagon, which is a peptide hormone, would regulate the gluconeogenesis in two methods. First, the covalent modification and the affection of transcription of the genes. How? Glucagon is released by alpha cells inside the pancreas. So, they would increase the transcription of the gene for PEPCK, leading to the increase in the rate of gluconeogenesis. Cortisol have the same effect of glucagon, by the way. The other method, which is covalent modification, is that glucagon would bind to G protein coupled receptors, leading to the increase of the cyclic AMP production and to the cyclic AMP dependent protein kinases. These dependent protein kinases will phosphorylate and inactivate the hepatic pyruvate kinase. Remember, pyruvate kinase was required during glycolysis for conversion of PEP to pyruvate. And so, when we inhibit this enzyme, PEP would not be converted to pyruvate. Instead, it would be used in gluconeogenesis. The second method for regulating gluconeogenesis is substrates availability. If the gluconeogenesis precursors are available, they would activate the process. Decreased levels of insulin would favor the mobilization of amino acids from muscle proteins to provide us with carbon skeletons from these amino acids. So decreased insulin would increase gluconeogenesis. ATP and NADH are important coenzymes for gluconeogenesis. They are provided by fatty acid oxidation, and so high concentrations of NADH and ATP would increase the rate of gluconeogenesis. As we mentioned before, acetyl-CoA activates pyruvate carboxylase. During fasting, tag hydrolysis would increase, and so fatty acids concentrations increase too. Fatty acids would produce high concentrations of acetyl-CoA, which finally activate pyruvate carboxylase. High levels of AMP activates phosphofructokinase. This means that the cell is derived from energy and required to increase the rate of glycolysis and energy production. So AMP would inhibit the gluconeogenesis. We have reached the end of our lecture, and I hope you found that the lecture was easy. And good luck.